Thank you so much for inviting me. It's really a great pleasure for me to be speaking with you today. And as you heard, for the past decade and a half, have been focusing along with other groups on studying the host microbiome interactome. In starting to speak to you about what we think and how we view this expanding field, I will start by highlighting four major hallmarks or hallmark concepts that have emerged in a decade and a half of microbiome research and will take us to more concrete examples throughout the talk. So the first concept is that while we mostly study the gut microbiome, which is one of the densest populations of microbes in our body, we need to realize that microbiomes exist at every site at our body that interacts with the outside world. And in each of them, they've evolved to supply important unique functions that are very unique to that particular niche. And I'll give you at least one example from our own work later on. The second hallmark concept is that if I were to characterize the microbiome in all of your guts or in all of your bodies deeply enough, I would find that each of you, and myself included, has a unique fingerprint of functional and compositional microbiome configuration. This was very scary to us at the beginning because we all like to reduce our studies and to be very uniform, but we've actually learned to harness this inter-individual variability towards understanding how the microbiome impacts different features of disease in people that harbor the same genetic risk factors. And I'll show you one example of our own work. The third hallmark concept is that we may regard the microbiome as a signaling hub that integrates into it signals that come from within our body, the host genetic system, immune system, and others, but also from a variety of lifestyle factors, such as the one that exemplified in the cartoon, that signal to the microbiome. The microbiome integrates all of these diverse signals, communicates them to the host, and we see one of our missions in life as decoding the letters of the alphabet of this very intense and intimate communication channel between us and our microbes. And finally, the past 3 or 4 years, the field has learned to appreciate that in addition to local microbiome effects on the human body, the microbiome may impact organs and cells that reside miles away from where the microbes actually live in. In many cases, this is mediated by thousands of small, potentially bioactive molecules, which we call metabolites, that can influx from where the microbes generate or modify them into every cell and organ in our body. And we see our interest as trying to contribute towards a better mechanistic understanding of host microbiome interactions. And if I were to break this down, we try to understand how the microbiome impacts human physiology, how the human body impacts the composition and the function of its different microbiomes, and what happens when the intricate communications between us and our microbes goes wrong that leads to a susceptibility to develop one of many multifactorial disorders. And by reaching such understanding, we hope and believe that we may be able to reach new principles that would enable microbiome-mediated disease treatment. And in this talk, I would like to highlight some vignettes of my 2 cents on where I think microbiome-mediated disease treatment is going to in human medicine. Now, I think it is fair to say that in 2021, when we speak about microbiome-associated treatment, we still focus on 3 very non-personalized and in many cases not sufficiently data-driven therapeutic approaches that include prebiotics, which are nutritional interventions, mainly made of fibers that are aimed at improving the health of a microbiome, whatever that means. Fecal microbiome transplantation that is exceedingly effective in Clostridium difficile colitis in the hospital setting, but it is less proven in other settings, and probiotics, which I will not go into today. But what's more encouraging for us is that with the amazing amount of data accumulating from microbiome studies, we are increasingly able to develop better science-driven or evidence-based microbiome approaches that I hope and believe would mature in the next decade of research. And I'll highlight 5 avenues using vignettes from our own work in the last decade. The first example of a microbiome-associated treatment that is under development is called personalized nutrition. Now, when we started looking into potential links between nutrition, the microbiome, and downstream host metabolic health almost a decade ago, I started reading all the quite boring literature that forms the basis to everything that you and I do in our daily lives when we try to come up with what we call a quote-unquote healthy diet. And what came to mind is that everything that we do is based on several grading systems that give different numbers or values to foods and food components. And based on these, we try to mix and match these numbers in order to come up with what we hope would be a healthy diet. For example, calories is such grading system. An even more widely used system that is at the basis of every diet that you've ever tried is called the glycemic index. And it is based on very small scale studies in the 1970s, which usually included 10 healthy individuals who were given an identical food, and then their blood sugar levels were measured by skin prick for 2 hours. So for example, if you were to take 10 healthy individuals, you would give them an identical piece of celery and measure the blood sugar level, you would see a small spike, just such as the one in the black graph, that would be denoted as an average number, and this would be the glycemic index of celery. Now, if you were to ask these 10 people to kindly eat an identical piece of chocolate cake, their average glycemic response would be higher. It would be denoted as the blue graph, and this would be given a higher number, which would be the glycemic index of chocolate cake. Now, if you were to look in your smartphone, you would find endless tables giving glycemic index values to any food and food component on Earth. And take my word for it, whatever diet you've ever tried, whether you went to a dietitian, a physician, you bought a book at the airport, is based in one way or another on this glycemic index system. Now, here comes the big problem. When we repeated this study design from the 70s, but rather than using 10 healthy individuals, we've used 1,000 healthy individuals, and we've given these 1,000 individuals one of several test foods, for example, 50 grams of glucose or an identical piece of white bread, and measure their blood sugar levels. We found that the average responses of these 1,000 people to every food was indeed the glycemic index of that food. So the system is inherently correct. However, when we looked at the individualized responses of these people when eating the same exact food, we found an unbelievable variability. Some people would drink 50 grams of glucose and would spike to the roof, and others would not care at all. And this was true for the test foods on the left. It is even more true for thousands of real-life foods that we've tested on the right panel. The variability was just unbelievable. And this already intuitively told us that this inherent system, assuming that there is a one-size-fits-all diet that would fit everyone, cannot exist, because if your glycemic response to a given food is opposite than mine, then the same diet cannot be good for both of us. And this eureka moment gave rise to a very ambitious study, which is called the Personalized Nutrition Project, in which we took these 1,000 people and kindly asked them to give us a week of their life, including many questionnaires, blood tests, a very big assessment of the microbiome, obviously, because this is what we do, anthropometrics, a smartphone app that we've developed for this project, which enabled to track in real life everything these people were doing during a week of follow-up, and a continuous glucose monitor that enabled to get an accurate glucose result every 15 minutes for an entire week. So in comparison to 10 healthy individuals in the 1970s, each giving 6 skin prick glycemic results, here we had 1,000 people, each giving us 2,500 accurate blood glucose measurements. But the interesting part happened after the week of follow-up, when a very smart group of students took all of this data from each individual and used machine learning and AI technologies in order to devise predictive algorithms that are aimed to predict a person's response to any given food, including foods they've never tried during the week of follow-up. And to summarize 6 or 7 years of work into one slide, these were the gist of the results. We've calculated the gold standard predictability when we go to a dietitian or a physician and we ask to be given a diet that would lower our blood sugar levels. There is some sense to the recommendation we get. We usually are prescribed a carbohydrate-poor diet. And the predictability of this kind of an approach is very low, it's 0.38. But when we use the data-driven, science-based, personalized approach, we dramatically improve our ability to predict a person's response to 0.68 in the middle panel. And in these kind of drills, you have to freeze your algorithm after proper training, apply to a fresh cohort. In this case, it was 100 individuals, which gave us the same predictability. Now, this was very reassuring, but it was now time to put ourselves to the real-life test. We've recruited a group of pre-diabetic individuals. These are individuals already suffering from glycemic problems, not yet full-blown type 2 diabetes. Just to give you a flavor, 40% of the American adult population is estimated to be pre-diabetic, 70% chance of developing signs of type 2 diabetes within the next decade of their life, and very little to offer them. So we took these pre-diabetics, we put them through our process, and this time we asked the computer to generate a set of personalized good and bad diet for each individual that are isocaloric, so not to blame the calories. And we asked each participant to follow their personalized good diet for a week, then their personalized bad diet for a week, while we extensively monitored them. And the diets were very counterintuitive, and a good diet could include some chocolate, a bad diet could include tomatoes. It was very surprising, and you cannot guess it until you measure it. But the fact of the matter was that a week of personalized good diet improved these pre-diabetic controls to almost normal glycemic responses, while a bad diet worsened them almost to diabetic responses. And when we visited the algorithms that are completely unbiased in retrospect and asked what they were using, we found many features related to the microbiome. In fact, today, after we've done hundreds of thousands of people, we can use just the microbiome plus a limited set of clinical features to reach a comparable accuracy. Of course, the billion-dollar question is whether we can put ourselves to the real-life long-term test. We've just completed and published our long-term study in which we followed 200 pre-diabetic individuals randomized into the American Diabetes Association recommended diet or to the algorithm diet, followed for a year. And you can see here an example of 3 independent glycemic control results over the year. You can see in red the mild improvement that has been noted before us with the one-size-fits-all recommended diet. And you can see in green that the personalized data-driven diet outperforms the American Diabetes Association diet, including a much higher compliance of participants. Now, this was an example of how we can harness diet and the microbiome towards the development of rational treatment in the future. A second avenue of treatment involves the transfer of either discrete microbes or whole communities. I've mentioned fecal microbiome transplantation, and I'll give you an example of our own work focusing on another microbiome, the vaginal microbiome, a very interesting, quite poorly studied microbiome that is usually dominated by lactobacilli species, but in a very common women's disease called bacterial vaginosis, it is replaced by a mostly anaerobic microbiome configuration that causes many symptoms, including smell, discharge, interpersonal problems, and so on and so forth. Now, most women respond by taking antibiotics, but there's a significant minority of women, around 10% of all women worldwide, so it's a huge number of affected individuals, suffer from what we call recurrent or intractable bacterial vaginosis that is no longer responsive to antibiotic treatment. And in addition to the usual symptoms, these women suffer from a dramatic increase in the risk of developing a number of obstetric and gynecological complications, such as the ones depicted here. So this is a huge unmet need, and we thought that maybe, just like fecal microbiome transplantation, that is very effective in reducing recurrent Clostridium difficile infection in the hospital setting, maybe vaginal microbiome transplantation could do the same trick in intractable bacterial vaginosis. So with collaboration with a group of fantastic gynecologists here in Israel, we've worked years to develop this system and to assure to the best of our ability the safety of this transfer. And once we were sure enough, we performed the first in-human pilot study in which we took a group of intractable bacterial vaginosis patients that suffered for decades without treatment. And we've performed a transfer from 3 healthy controls. And this is the gist of the result. You can see in the y-axis, the Amsel criteria, which are the severity of bacterial vaginosis criteria. You can see that all of these participants suffer from the worst Amsel criteria with spikes appearing every few days. This is how they live. And once a successful vaginal microbiome transplantation had been performed, all of these patients went into complete remission. We now follow them for 3 to 4 years, and we don't see relapses. What was even more interesting is what we saw when we sequenced their vaginal microbiome before and after the procedure. You can see here a principal component analysis graph in which each dot represents one sample. And you can see that the 5 full red samples of the pre-vaginal microbiome transplantation patients, these intractable bacterial vaginosis patients are clustered very far apart from the three full colorful circles representing their three healthy donors, meaning that the microbiomes of the diseased and healthy individuals were very distinct from each other. But what's most fascinating is what happened after a successful VMT. And you can see the arrows leading from the red dots into their new configuration, which is clustered very closely to that of the healthy donors, meaning that in addition to the clinical remission, the microbiomes were reset back to healthy configurations. And now we're engaged in much larger controlled trials to see whether we can improve our knowledge and maybe offer this as a future microbiome transfer treatment. The third avenue of microbiome treatment relates to these small bioactive metabolites that I've mentioned at the beginning of my talk that are either generated or modified by the microbiome. And if we can understand discretely how each of these small molecules signals to the host, we can replenish it when it's missing or block its signaling when it's abundant in a disease, and offer treatments that would bypass this entire microbiome inter-individual variability that is so limited. And this is called postbiotic therapy. Just to give you one example of our own work, we've recently got interested in a deadly neurodegenerative disorder called amyotrophic lateral sclerosis, or ALS, a disease which manifests as motor neuron weakness that progresses until people die within 3 to 5 years, absolutely no cure. So again, a very big unmet need and a lot of variability between patients, which always leads us to think that environmental factors and maybe the microbiome could play a modulatory role. So we started by exploring this in the most widely used mouse model for carrying the gene that specifies the most common form of familial ALS, the SOD1 transgenic mice. And we used many different readouts. I'm showing you for the sake of time 2 major readouts, the Rotarod test and the grip test, which are objective readouts in which the higher the score, the stronger the mouse, the lower the score, the weaker the mouse. And you can see in blue that the group of genetically susceptible ALS mice gradually develop progressive weakness in both tests. But interestingly for us, when we depleted the microbiome of these mice at our facility, this is the red group, the disease became much more progressive and much more severe in the absence or depleted microbiome. This was also the case when we rederived these SOD1 transgenic mice to the germ-free setting, meaning that something in our microbiome at our facility was modulating disease course. And this led to a very extensive shotgun metagenomic sequencing quest in which we identified 11 strains that are associated with changes throughout disease progress at our facility. 10 of them seem to be associated with a worsened disease course. One of them was associated with an improved disease course. So this became the interesting bacteria for us, and its name is Akkermansia. And to prove that it's not just correlated, but actually does something, we replenish the missing Akkermansia from SOD1 transgenic mice. And you can see here an example for one of the experiments in which, in this case, the control SOD1 transgenic mice were in red, and you can see the progressive weakness that is induced spontaneously throughout time in this model. And mice that were identical but were replenished with Akkermansia, with this missing bug in black, featured an ameliorated disease course, meaning that somehow Akkermansia was modifying disease towards a positive, more positive outcome. And of course, the next question was, how does Akkermansia do what it does? And this resulted in a very extensive metabolomic study, a mass spec-based study that aims to characterize the differentially expressed small molecules in the many different conditions that were tested in this study. And we came to the finalist 27 metabolites that were differentially expressed. And basically, we focused on one of them, the most promising candidate, which is called nicotinamide, which seemed to be depleted in mice that developed ALS and was overexpressed in mice that were supplemented with Akkermansia. And to prove whether it does something, we supplemented this missing metabolite by osmotic pump to SOD1 transgenic mice, and of course compare them to controls. And to make a long story short, we found that in contrast to the red control SOD1 transgenic mice that developed the typical deterioration in ALS, mice that were supplemented with the missing metabolite were improved and were slower to progress. We also proved that this metabolite was actually secreted by Akkermansia, and it reached the central nervous system by influxing from the gut to the systemic circulation through the brain-blood barrier into the cerebrospinal fluid. And finally, we wanted to scrape the surface and see whether this concept that we've stumbled upon may hold true in humans. So we've assembled a cohort of around 100 ALS patients and household healthy control. We sequenced their microbiome. We found many different changes. We performed systemic metabolomics, in which we characterized the thousands of small molecules in their blood, and we found a huge difference in the signatures between patients and controls. And we even, in a subset of patients, were able to do this in their cerebrospinal fluid. And interestingly, both in blood and in the CSF, we found that this nicotinamide metabolite that was identified in mice was suppressed and was reduced in its quantity in ALS patients as compared to controls. And obviously, you may guess that there are many follow-up studies in humans and in mice that have been published and are being conducted in trying to see whether we can harness the microbiome and its associated metabolites towards some sort of treatment or amelioration in this deadly disease. The fourth avenue of microbiome treatment relates to a huge unmet need. Imagine that you would find that the commensal bacteria, wherever, is associated with disease pathogenesis and with disease progress. How do you get rid of a bacteria in the microbiome is a huge unmet need. Antibiotics are a very bad way to do this. They're non-specific, they're associated with adverse effects, with the emergence of resistant strains. So you cannot give antibiotics forever. And really, we have no solution on how to do this without impacting the entire critically important microbiome that is around these pathobionts. One way that we try to solve this is through the use of bacteriophages. Bacteriophages are these special viruses which only can infect bacteria. There is no receptors known on eukaryotic cells. And therefore, phages could attack and destroy bacteria. The problem is that bacteria have evolved many different defense mechanisms against phages. For example, CRISPR restriction enzymes are all tools that bacteria developed to fight phages. So there's a huge arms race between the two kingdoms. And so to avoid this resistance developing as a therapeutic modality, we decided to generate phage cocktails in which each component targets a different receptor in a different pathway in the attacked bacteria, and together the cocktail would win and would enable to kill the bacteria without the development of bacterial resistance against phages. So we've generated this factory of thousands of phages, which we genomically characterized. And in a first attempt to see whether we can use this system, we focus on Crohn's disease. We've assembled 4 cohorts from around the world. We've extensively sequenced them for their bacterial gut microbiome. And we found some common denominators that could represent pathobionts that may contribute to the pathogenesis of Crohn's disease across these biogeographical locations. We focused on one of them, Klebsiella, and we developed a 5-phage cocktail against the IBD-associated strains of Klebsiella that we've identified across these 4 international cohorts. We've tested this cocktail of phages in a number of preclinical models. You can see here an example on the right panel in which the administration of some Crohn's disease-associated human Klebsiella strains into germ-free mice induce a Th1 response, which manifests as an autoinflammatory disorder, which mimics Crohn's disease. And once you give the phages that eradicate or deplete these bacteria, you also deplete the inflammatory response and disease severity. And we've even went forward to a phase 1 clinical trial in which we generated a human-specified phage cocktail against the same IBD Crohn's disease-associated commensals. We've given it to a group of healthy volunteers. It seemed to be safe, and it accumulated at a dose that was 1,000-fold higher than its bacterial target, which is what we aim for. And now we are in midst of phase 2A clinical trials in Crohn's patients, seeing whether we can do something to these pathobionts and whether we can do something to disease severity. And finally, I would like to finish with the last avenue, which is an avenue which we tend to forget, which consists of the host. And the host is a very integral and important part of host microbiome interactions. And by understanding these interactions, we are bound to find new therapeutic targets in the host. And just to give you one final example of our own work, we've recently focused on another deadly disease in humans called acute liver failure, or ALF, which is a devastating and fulminant disease affecting both adults and children. In both, the most common cause of acute liver failure is drug-induced hepatitis, and mainly in the developed world, acetaminophen or paracetamol intoxication. And once a person develops this rapidly progressing failure, that person has an 80% chance of dying unless an urgent liver transplantation is allocated. So we wanted to understand some of the more molecular and cellular levels driving acute liver failure. So we started with mice with two different models of drug-induced hepatitis. One of them was acetaminophen. Another one was inducing a more autoimmune phenotype. And we performed single-cell transcriptomics in tens of thousands of liver cells before and after the induction of acute liver failure. And to make a very long story short, we stumbled across three new cell states or cell types, Kupffer cells, endothelial cells, and stellate cell subsets that have never been described to date and only seem to arise during the development of acute liver failure in these mice. Now, when we looked at the genomic repertoire of these three new cell states, we were surprised to find that all of them seem to be enriched and to be potentially controlled by genes that are regulated by the transcription factor MYC. And this was not a coincidence. We made sure that this was the case. However, this was an interesting observation and correlation and not causative. And since we pride ourselves on trying to reach causation, we repeated these acute liver failure induction studies in mice, now adding a known competitive MYC inhibitor to the system. And indeed, in all the different measurements of liver failure that we've tested, for example, immune infiltration, the elevation of serum liver enzymes, histology, and mortality, the inhibition of MYC seemed to markedly improve disease severity. And when we performed single-cell transcriptomic of these MYC-inhibited or not inhibited mice, we again looked at this MYC regulated signature, which consists of 88 different genes. You can see here an example of one of the experiments. On the top, you can see the 88 genes. On the bottom, you can see an example of one of these genes, CCL2, and you can see that during health, these genes are not expressed in any of the three cell types that we've discovered. So there's absolutely no expression. Once you induce acute liver failure, the expression levels of these genes jumps to the roof, no matter which acute liver failure model you decide to use. And once you inhibit MYC, again, the expression of this MYC hallmark gene landscape is completely suppressed. Now, what does the microbiome have to do with this? We tested this first by depleting the microbiome and inducing acute liver failure, or doing this in germ-free mice. And what we found was that in the absence of a microbiome, disease was markedly attenuated in all mouse models that we've used. And then we performed single-cell transcriptomic analysis of, in this case, germ-free or colonized mice, but we did this also with antibiotic-depleted mice and looked at the changes. And I'm showing you here two of the three cell subsets that we've discovered. The third behaves the same. And you can see in the y-axis, the gene expression profile of colonized mice, in the x-axis of non-colonized mice, both of them induced with acute liver failure. And when we looked at the 82 genes that are controlled by MYC, there is a much higher expression in colonized conditions as compared to germ-free conditions, in parallel to the improvement in disease severity in the absence of a microbiome, suggesting that the microbiome controls as an upstream set of signals this MYC signature. Now, the next, of course, question is, how does this happen? The obvious suspect is a very well-known pathway controlled by TLRs, which signals to a set of very well-known adapters upstream to MYC. And these TLRs could be activated by microbe-associated molecular patterns coming from the microbiome, or by damage-associated molecular patterns coming from the damaged hepatocytes. And to prove that this is indeed the responsible pathway, we knocked it out, including genomic or pharmacological inhibition of each and every one of its known adapters. And to make a long story short, we found that the system results in microbiome plus damage hepatocyte activation of one branch of this toll-like receptor pathway, leading to p38 activation and then to MYC activation downstream to this pathway. Now, finally, we wanted to again scratch the surface to see whether this may be potentially relevant in humans. So we've allocated from the leading tertiary transplant center in Israel liver samples from 7 acute liver failure patients that were transplanted, and as controlled, we used their healthy, quote unquote, because they were cadaveric donors. And when we stained for MYC, we found a really stark difference between the acute liver failure samples, which seem to have a diffused activation of MYC as compared to no activation in the healthy livers. And this, of course, leads to continuation studies focusing on mechanism, also in the human setting. So to summarize this story, we found a new microbiome and host-regulated pathway in the liver that is completely shut off during health, but turns itself on during the induction of acute liver failure, leading to the emergence of 3 new cell states, which secrete massive amounts of cytokines and chemokines, which exacerbate and lead to full-blown acute liver failure. And importantly for our story, when we inhibit either the microbiome or distinct checkpoints along this pathway, we can ameliorate this disease, at least in mice. So to summarize my talk, I try to convince you that one of the biggest challenges and exciting prospects of the microbiome field is to use a lot of the data that is being generated and to use methods that are aimed at reaching causality in order to develop a new microbiome-based treatment and to harness them into the evolving precision medicine landscape. And I hope and believe that in the next decade, you will start seeing these on your clinical shelf. And I'll end by thanking the many people at my Weizmann Institute group who came from all over the world and are engaged in these really ambitious and exciting, I think, studies, our collaborators, the funding agencies that are making our work possible. And in the last year and a half, also our cancer and microbiome division at the DKFZ, a fantastic group of researchers who try to apply all the know-how and technologies that we've previously developed towards the study of the microbiome in the context of cancer development, progression, metastasis formation, and responsiveness to treatment. And with that, I'll stop here and I'll be happy to try and answer some questions. Thank you.